Bismillahirrahmanirrahim. Assalamu alaikum. Hello and how are you all? And welcome to Biosciences with Shatreen Sawati. And Shatreen Sawati is here to deliver the lecture from my coronavirus series. But first of all, Ramadan Mubarak to all Muslim Ummah. And I have lots of prayer for all the Muslims. May Allah protect all of us from this coronavirus. So today I'm going to deliver my next lecture about the coronavirus, and in this lecture I will discuss about the molecular targets of the coronavirus. As I have discussed all the molecular targets in my previous lecture, but not in detail. In this lecture, I will discuss all these molecular targets in detail, like ACE2, transmembrane proteases, serine proteases, and furin and neuropilin. So, I have a very humble request to all of you. If you did not subscribe my channel, subscribe my channel and like my videos and share my videos. If you are sharing my videos, you are sharing the knowledge. And I'm so happy to know that you are getting benefits from my lectures. And I got very good feedback from all of you. And kindly do promote my channel. Not only my channel, but all the educational channels, all the educational contents, do viral, do promote them. So now I am starting my lecture about the molecular targets of the SARS coronavirus. In this lecture, I will discuss about the nature, location, functions, and role in SARS coronavirus infection. If you people did not watch all the students, research students especially, who are working with the coronavirus and want to do the research, if you did not watch all my lectures, watch my lectures, and if you have any question, kindly share with me. If you need any help, I will try my best to solve or to resolve your problem. So this lecture I created on the basis of all the requests related to the molecular targets. So I hope so the lecture will be clear upon you people, and you can easily get more about the molecular targets of the coronavirus. So must watch my lectures about the life cycle and especially spike protein and structure of the coronavirus, and then this lecture will be easily understandable for you people. I will not discuss in detail about the life cycle and many more. I just focus on the molecular targets. So first of all, I will give you the introduction about the virus and host interactions. Virus host interaction determines cellular entry and spreading in the tissues. Severe acute respiratory syndrome, coronavirus 2, and the earlier SARS coronavirus use angiotensin converting enzyme 2 as a receptor. So first molecular target I will discuss here is the angiotensin converting enzyme, means ACE2, which is the main receptor for the coronavirus. Okay. Now a little bit I'm going to give you information about the how do S proteins become active. I have discussed in detail in my lecture about the S proteins, so must watch my lecture about the S protein. You will get a lot. S proteins are the spike proteins, and these spike proteins help in the attachment of the infection of the coronavirus. So in the native state, the coronavirus S protein exists as an inactive precursors. They exist as an inactive precursors when the coronavirus do not interact, do not enter into the host body, like the human body. During viral infection, target cell proteases activate the S protein, and I will discuss in detail about the protease activators. So the protease activate the S protein by cleaving it into its subunit S1 and S2 subunit. S1 subunit help in the attachment of the fusion of the coronavirus with the host cell. So must watch my lecture about the life cycle and the S protein. You will get a lot, which is necessary for activating the membrane fusion domain after viral entry into the target cell. Is it clear? So the spikes help in the attachment or fusion of the coronavirus, and as well as in the help in the infection, or you can say the entry of the coronavirus into the host or human cell. Similar to the other coronavirus, the S proteins of SARS-CoV-2 is cleaved into S1 and S2 subunit by cellular proteases, furin, and the serine proteases, transmembrane proteases, which is serine proteases, is used as a protein primer as well. So I will discuss all these things step by step, all these molecular targets step by step. Now you can see here the clear figure of the coronavirus, and this is the coronavirus, and this coronavirus come and attach with the human cell, and at the human cell membrane surface there are receptor present. One is which I'm going to discuss is the ACE2 angiotensin converting enzyme. This is the main target for the coronavirus, and there is another proteases which is known as transmembrane serine proteases, which help in the cleavage of the spike protein into its subunit. As I have discussed that the spike proteins present in inactive form when it do not come in contact with the host cell. So after when the coronavirus come and attach with the SU2, then what will happen? The transmembrane proteases activate it by the cleavage of the S protein into its subunit S1. And you can see here the these transmembrane proteases act as a you can say scissor for the cutting of the you can say that responsible for the cleavage of the S proteins or spike protein into its subunit S1 and S2. Okay. Now what is the ACE2 receptor? I got lots of questions about the ACE2 receptor. You can get lots of information from the research article, and I created this and all my lectures on the basis of the research. So I will give you some information ACE2, and you can also read from all these research articles just from the Google Scholar. You can get and read them. So now I'm just providing here the basic concept of the ACE2. ACE2 is a protein on the surface of the many cell types. These are the proteins, membrane proteins. Simply, you can say that these are the membrane proteins. It is an enzyme that generates small proteins by cutting up the large protein angiotensinogen that then go on to regular functions in the cell. Is it clear? Using the spike-like protein on its surface, the SARS coronavirus binds to the ACE2 like a key being inserted into the lock prior to the entry and infection of the cell. Hence, ACE2 acts as a cellular doorway, a receptor for the virus that causes COVID-19. So must watch my lecture about the life cycle of the coronavirus. You will get a lot. Okay. Now you can see here the ACE2 how interact with the coronavirus come and attach with the ACE2, and how the transmembrane serine proteases help in the cleavage of the inactive spike proteins into its subunit S1 and S2, and in this way the coronavirus enter into the host cell by endocytosis. And this is the simple way, and this diagram clear the concept of the SU2, which is the site for the attachment of the coronavirus, and the transmembrane proteases. Okay, what is the normal role of SU2 play in the body? Now this is very important. You must know that what is SU2. Now the concept clear. This is a protein. This is an enzyme which is present on the surface of the cell, or you can say this is a membrane protein. So what what is the normal role? Now, ACE2 is a vital element, is a vital element in a biochemical pathway that is critical to regulating process such as blood pressure, wound healing, and inflammation, called the renin angiotensin aldosterone system or SARS pathway. You are familiar with SARS pathway. If you are not familiar with the SARS pathway, must get information about the SARS pathway. Okay, so this is the normal role of SCE2. Now, angiotensin 2, what is this? SCE2 will help modulate the many activities of the proteins called the angiotensin 2 that increase blood pressure and inflammation, increasing damage to the blood vessel linings and various types of the tissue injury. Or simply you can say that the angiotensin play a role in the various types of the tissue injury. SU2 convert the angiotensin 2 to other molecule that counteract the effects of the angiotensin 2. So SU2 play a very important role in the conversion of the you can say the harmful activity of the angiotensin 2 by converting into the types of the molecule and reduces its effect, or you can say that reduces the injury to the cell. Of general relevance to the COVID-19, angiotensin can increase inflammation and the death of the cell in the alveoli, which are critical for bringing oxygen into the body. These harmful effects of the angiotensins are reduced by the SU2. So here I'm giving you the concept of the SU2 and angiotensin 2. Angiotensin 2 is a type of the proteins which you can say that causes the injury to the tissues. So SU2, what will happen? The SU2 convert the angiotensin 2 into other types of the molecule. I'm not discussing here those molecules, but just giving you a concept that the angiotensin converting by the SU2 into other type of the molecule and prevent the cells from the injury. So what will happen if the coronavirus will attach to the SCE2? Okay. When the SARS-CoV-2 binds with SU2, it prevents SU2 from performing its normal function. Yes. If the coronavirus attached with the SU2, what will happen? The SU2 function will be interrupted. So, performing its normal function to regulate angiotensin signaling. Is it clear? Thus, SU2 action is inhibited, removing the brake from the angiotensin signaling and making more angiotensin available to injured tissues. The decrease, you can say that the decreased braking likely contribute to injury, especially to the lungs and heart in COVID-19 patients. That will sometimes leads to the death of that patient because of that activity. Now, I hope so the concept will be clear upon you people. Okay. Now, which organs are most severely damaged by the SARS-CoV-2? Most of the people ask that which organs are severely damaged by the SARS-CoV. So, if all those organs which have SU2 and transmembrane proteases can be affected by that. So I'm giving you here, the lungs are the primary site of the injury by SARS-CoV-2 infection. The virus also impact other tissues that express SU2. So all those tissues, all those parts of the body which expresses SU2 enzyme, SU2 proteins are easily infected by the virus. So, including the heart, where damage and inflammation can occur, the kidneys, the liver, and the digestive tract can also be injured. Blood vessel may also be site for the damage. So it will be clear upon you people that how it will affect and which part of the body are affected by that, and what is the reason behind it. That ACE2, which is the angiotensin converting enzyme, the protein, which is and the other transmembrane proteases, which are present in abundant amount in the cell membrane of all these, or you can say express here in all these types of the cells of all these organs. So all these organs are affected by the coronavirus. Is it clear? Okay, now you can see here clearly all those organs where the ACE2 express, and where there is ACE2 there is a chance of infection of the coronavirus. Is it clear? Okay. Now transmembrane serine proteases, I'm going to give you the concept of transmembrane serine proteases. As I have discussed that transmembrane serine proteases act as like a scissor, which help in the cleavage of the what? Spike protein into its subunit S1 and S2. And now when the spike protein cleave into its two parts, into its subunit, then the coronavirus will be able to infect the human cell or host cell. Okay, what is now the transmembrane proteases? Transmembrane serine proteases 2 is a cell surface protein primarily expressed by the endothelial cells across the respiratory and digestive tract. As a serine protease, it is involved in the cleaving peptide bonds of the protein that have serine as the nucleophilic amino acids within the active site. Now, the concept clear about the why it is called serine protease, because the serine as the nucleophilic amino acid within the active site. The exact biological function of the transmembrane serine protease is largely unknown, although research has shown that it is involved in the certain pathologies. So I am not discussing here all these pathologies here. So transmembrane proteases are expressed in the nasal, bronchial, and gastrointestinal epithelium, and you can see here there is ACE2, and where there is ACE2 you can see here the transmembrane serine proteases, and this transmembrane serine proteases act like a scissor and do the cleavage of the spike proteins into its subunit, and then the coronavirus will be able and become active after the cleavage of the spike protein, and then causes the infection to the host. Okay, I hope so the lecture will be clear upon you people. I will discuss the furin proteases and neuropilin proteases in my next lecture. Thank you very much. Till then, Allah Hafiz.